Hello, everybody. My name is Noham Wolpe from Tau, and I got the honor of chairing this penultimate session. This is going to be another excellent session with two, not one, but two BIRACS awardee projects. But before we get to hear about these projects, we're going to hear from Melody Kasher. Speaking of awards, she got the Herteg Institute on Aging award last year, was it 2020, for an excellent, excelling PhD student. So looking forward to hearing your talk. And this would be on shared genetic architecture between rheumatoid arthritis and varying osteoporotic phenotypes in the aging population. Thank you.
So, hi, my name is Melody Kasher. I'm a PhD student at Tel Aviv University under the supervision of Professor Livshits. And my presentation is about the shared genetic architecture between rheumatoid arthritis and varying osteoporotic phenotypes, two musculoskeletal conditions that manifest in the aging population. So the research entails two highly prevalent, debilitating, and degenerative musculoskeletal conditions, rheumatoid arthritis and osteoporosis, which plague the aging population and contribute to a high burden of disability. As can be seen on the graphs on the right, there's high incidence in the aging population for both diseases. A little bit about rheumatoid arthritis. It's an autoimmune condition. Susceptibility increases with aging. Females are at a greater risk than males, at a ratio of 3:1. The prevalence is approximately 1% globally. It's multifactorial and associated with osteoporosis. Osteoporosis means porous bone. It's the gradual degeneration and deterioration as aging occurs. It's predominantly observed in females like rheumatoid arthritis. Bone mineral density, which serves as the main predictor of low trauma bone fracture, is commonly accepted as a key measurement in several skeletal sites in the development and diagnosis of osteoporosis. So as I mentioned, these diseases are both multifactorial, so as well as having an environmental component, they have a genetic component. Rheumatoid arthritis has a heritability rate of approximately 60%, while osteoporosis has a heritability rate of at least 50%, but it depends on the skeletal site in question. So in our lab, we previously assessed the relationship between rheumatoid arthritis and various osteoporotic markers in this Kazakh sample. The most outstanding observation was seen in the middle graph in the bone mineral density, where the rheumatoid arthritis affected subjects, which are in the blue, have a much lower bone mineral density compared to the unaffected counterparts in each age group. So this was in the Kazakh sample, which has 405 clinically diagnosed rheumatoid arthritis patients and 397 unaffected subjects. So this result is clearly indicative of the occurrence of bone deterioration during the early stages of rheumatoid arthritis, followed by the progression of a similar rate seen in the unaffected population. So we also looked at the ELSO data set, which stands for the English Longitudinal Study of Aging. And this data set consisted of approximately 10,000 subjects, where 628 of them had rheumatoid arthritis. And we were looking at risk factors of rheumatoid arthritis, and we see that those with rheumatoid arthritis have a higher age, increased BMI, HSCRP, which is a high sensitive C-reactive protein. It's an indicator of general inflammation and used to suspect rheumatoid arthritis, and osteoporosis as well. So this led us to our two major research questions. The first question was, assuming a genetic relationship may explain the association between rheumatoid arthritis and osteoporosis, is the nature of the genetic relationship between rheumatoid arthritis and osteoporosis pleiotropic, or perhaps related to other contributing factors or underlying mechanisms? So there are three kinds of pleiotropy that are generally examined. One is mediated pleiotropy, which can be examined by Mendelian Randomization, which looks for causality, and this is when one genetic variant contributes to the manifestation of one disease, which causes the other disease. There's biological pleiotropy, where shared common variants or distinct common variants of the same gene contribute to the manifestation of both diseases. And then there's spurious pleiotropy, which is that the genetic variants responsible or contributing to both diseases are actually in different genes that are in linkage disequilibrium. And then our next question was, since rheumatoid arthritis, an inflammatory condition, necessarily contributes to raise C-reactive protein, and this likely leads to osteoporosis through bone turnover mechanisms, does CRP genetically influence the relationship between rheumatoid arthritis and osteoporosis? So genetic statistical tools were carried out to clarify the genetic nature of the relationships and potential causality using big data resources. We use Mendelian randomization to look for causality between exposure and outcome variables. We use colocalization to test for pleiotropy, and we use genetic enrichment analysis to identify the genetic variants that were isolated. So our data sets include several data sets from various resources, but the biggest data set we used was the UK Biobank, which is made of 500,000 subjects. And in that, we have 5,000 subjects with rheumatoid arthritis, 7,000 with osteoporosis, and about 250,000 subjects with bone mineral density measures of the heel using ultrasound measures, whereas the other ones use DEXA, which is dual X-ray absorptiometry, and have different skeletal sites here. So Mendelian randomization, like I said, it tests for the causal effect of proposed exposure on the outcome, and it is dependent on 3 assumptions. The first assumption is that the instruments or genetic variants must be associated with the exposure. The second one is that the instruments or genetic variants must not be measured or unmeasured, must not have measured or unmeasured confounders. And the last one is that the instruments must influence the outcome only through the exposure. Otherwise, if this is violated, then it's grounds for pleiotropy, biological pleiotropy, which is also called horizontal pleiotropy. So in assessing Mendelian randomization, we use the MR-Egger approach, which provides an MR-Egger estimate, and which can be examined by the p-value. And it also uses the MR-intercept, which assesses horizontal pleiotropy. So when we're looking at rheumatoid arthritis, if it causes osteoporosis through any of these osteoporotic phenotypes, we see that generally there is no grounds for causation. However, we see that there's horizontal pleiotropy between rheumatoid arthritis and osteoporosis or bone mineral density. In the end, we see rheumatoid arthritis and C-reactive protein, and we see that rheumatoid arthritis is significantly causal for C-reactive protein without any evidence of horizontal or biological pleiotropy. Next, we looked at C-reactive protein and if it is causal for osteoporotic phenotypes or bone mineral density, and we saw that CRP is not causal for osteoporotic phenotypes. However, there's grounds for horizontal pleiotropy. So next, we conducted colocalization and gene enrichment analyses. We examined SNPs, or genetic variants, that appear to colocalize either through mutual common SNPs or distinct SNPs within the same gene. So colocalization analysis uses Bayesian modeling and has 5 hypotheses that are measured through posterior probabilities. And we were interested in hypothesis 3, which measures distinct common causal SNPs, or hypothesis 4, which measures mutual common SNPs to both diseases. And if we have a posterior probability of 75% or higher, this is strong evidence of pleiotropy, and 50 to 75% is moderate evidence of pleiotropy. Finally, we use functional mapping and annotation, FUMA, GWAS platform for gene enrichment analysis to define the genes in common between the 2 phenotypes. And this, again, is just a graph of the different kinds of pleiotropies that I previously described. So here we see, on the first column, we see the osteoporotic phenotype in question, which is colocalized with bone mineral density. With rheumatoid arthritis. And then basically in the last column all the way to the right, we see posterior probability of H4, which is shared common SNPs between the 2 phenotypes. And we see here that we have high evidence of shared SNPs between the 2 phenotypes. And then in the column in the middle with gene symbol and SNP, we see the genes that are shared. The same with CRP and osteoporosis, the same idea. Again, we have strong evidence of shared SNPs, and we see them in the middle. However, with rheumatoid arthritis and CRP, we have distinct common SNPs within the same, distinct causal SNPs within the same gene, and that's also shown here. I'm going a bit fast for the sake of time. And finally, conclusions and implications. Pleiotropy have independent, we suspect pleiotropy, and we are showing here that pleiotropy is responsible for the relationship between rheumatoid arthritis and osteoporosis, as well as C-reactive protein. Basically, rheumatoid arthritis and osteoporosis have their shared genes, shared SNPs, and then rheumatoid arthritis is causal for CRP, and at the same time, CRP and osteoporosis also share their own SNPs. And this would also lead to possibly the idea of inflammaging because of inflammation and rheumatoid arthritis and osteoporosis. And thank you.
Thanks. Unfortunately, we don't have time for questions.